good evening, and welcome to our second Friends of NSM Distinguished Lecture Series, or lecture of the series, and welcome to the University of Houston. It's nice to have you here. I'm Mark Smith, the Dean of the College of Natural Sciences and Mathematics. The Friends of NSM is a new initiative for our college where we try to engage the community and to build connections between our faculty, our students, our friends, and the scientifically interested community. So this is the number 2 talk. This will be the last of this semester. We have 3 more in the fall in the series of science impacting health and society, and I think there's some flyers for the next talk floating around. Let me go ahead and get started. I'd like to introduce Dr. Robert Schwartz, who will introduce today's speaker. He has kindly agreed to introduce our speaker tonight. And Dr. Schwartz is the Hugh Roy and Lily Krantz Cullen Distinguished Professor of Biology at the University of Houston's Department of Biology and Biochemistry. Dr. Schwartz is widely recognized for his research in gene regulatory aspects of embryonic heart development. In his field, he's received 13 U.S. patents and co-founded 3 companies. Prior to joining UH, he was the director of the Institute of Biosciences and Technology for 5 years and was a member of the faculty at Baylor College of Medicine with appointments in molecular and cellular biology and medicine departments. Dr. Schwartz is also a member of the Texas Heart Institute and director of its stem cell engineering laboratory. He has more than 200 reviewed publications dealing mostly with heart research and is a longtime friend and collaborator with Dr. Eric Olson. Dr. Schwartz.
So this is an enormous pleasure to introduce Eric Olson. Everyone has a handout. If you don't have a handout, you should have it. And you can look at the accomplishments of Eric Olson. It tells you the chairs, the Willie Nelson chair, how many chairs he has. There's at least 3 endowed chairs, and probably the most significant one is the Willie Nelson chair. And I guess it's in his band. He has a great band and stuff. So you can see all the wonderful accomplishments that he has done and has done in his career. So you can look at that. So basically, this is sort of like an extemporaneous talk about Eric. I knew Eric when he was an assistant professor at MD Anderson, just starting out, and we talked about research, and it was clear at that time that he was a force to be reckoned with. He moved so quickly through the fields and also to becoming one of the younger chairs of biochemistry at MD Anderson was astonishing. And every week there was something new coming out of Eric's lab, and it was just an exciting period of time. So Eric started off in Houston. He's our Houston boy. He's probably one of the great scientists in this town ever produced. And I at least had the opportunity of doing a program project with Eric for 15 years, and that was a wonderful period in my life in being able to work closely with Eric, and our friendship has continued on for many, many years. He's good friends with many of the people in the audience, especially from the medical center, that have come to hear Eric speak. It is a tremendous privilege and honor to introduce Eric Olson. He's one of the best in our generation, a member of the National Academy, and he does research at the highest levels. Eric Olson.
Thanks, Bob, for that introduction, and thanks for leaving out a lot of the details that you warned me about in the beginning. So I'm really happy to be here. I want to thank Dean Smith and Bob Schwartz for inviting me. I love Houston. This is where I grew up scientifically. I have many friends here, as Bob said, and I'm very happy to be back here to talk to you tonight, and I thank all of you for coming out this evening. So I'm going to talk tonight about 2 recent subjects of interest of our group. that focus on the heart, and I'm going to try to relate these to the theme of this series of lectures, which, as I understand it, is centered on biology and medicine. Our group, for 25 or so years, has used muscle cells as a model system to understand basic principles of developmental biology and gene regulation. And along the way, we've deciphered many of the regulatory mechanisms that control the formation of the three major muscle cell types: skeletal muscle, cardiac muscle, and smooth muscle. One of the unanticipated outcomes of this work is that as we began to unravel the basic mechanisms of developmental biology in these cell types, we came upon mechanisms that we think are fundamental in disease, and realized that many developmental mechanisms are redeployed in adulthood as the basis for disease, and that then opens opportunities for therapeutic intervention. And I'm going to give you two examples tonight in which developmental mechanisms can be leveraged to address major problems in human disease. This is a painting that I like by Paul Klee, and I'm going to begin by talking about the theme of this painting, that is the sick heart, and what we've uncovered recently about the mechanisms for the sick heart and some therapeutic approaches for intervention. Heart disease, as most of you are aware, is the number one killer of mankind. It's really humbling to realize that a myocardial infarction occurs every 25 seconds in the United States. Half of those are fatal. One of the ultimate outcomes of a myocardial infarction is heart failure, which is simply an inability of the heart to pump an adequate amount of blood to meet the demands of your body. 5 million Americans suffer from heart failure, and the incidence of heart failure exceeds that of all combined cancers. This is truly a major epidemic with a major need for new insights and new therapeutic indications. Now, there are approaches for treatment of heart failure, and many of these are familiar to you. The primary cause of heart failure is occlusion of the coronary artery, which feeds this region of the heart, the left ventricle, with blood. Heart muscle cells are exquisitely sensitive to the level of oxygen, and so deprivation of a region of the heart of oxygen, even for a short period of time, can have devastating consequences, as we'll talk about. There are several therapies currently available for individuals with heart failure. Beta blockers are certainly widely used. The most effective therapeutic intervention for heart failure would, of course, be a heart transplant, but it's impossible to meet the needs of the millions and millions of individuals who need new hearts. And there are also many biomechanical devices, such as left ventricular assist devices, LVADs, which Vice President Dick Cheney had, and many you may have heard about. Just for those of you who are not familiar with the heart, one slide of some basic cardiology 101. This is a cross-section through a heart. This is the left ventricle of the heart. That's the pump of the heart that pumps blood out to the rest of the body. The right ventricle pumps blood to the lungs. When the heart is injured by a myocardial infarction, it's the left ventricle that ultimately suffers with the loss of heart muscle cells, and that causes a resulting loss of pump function. This slide illustrates the problem that I want to talk about in the first half of this lecture. This is a cross-section through a heart. This is the heart of a mouse, but a heart of a human would look essentially the same. Here's the muscular wall. Here's the left ventricular chamber where the blood comes and is pumped out. Here's the right ventricle, which is smaller, and these are the two atrial chambers. When the coronary artery is occluded from blood flow to the left ventricle in a myocardial infarction, up to 25% of the several billion heart muscle cells in the wall of the left ventricle can die very quickly, and they're replaced by a fibrotic scar. And the process of fibrosis and scar formation impedes heart repair and obviously impedes cardiac contractility because the scar is a non-contractile tissue. And the fundamental problem in adult cardiology is the fact that adult mammals, including humans, cannot regenerate new heart muscle cells following an injury. Now, the process of organ regeneration has been the focus of interest for thousands of years, dating back to the ancient Greeks, who recognized that the liver has the ability to regenerate, as documented in the ancient myth of Prometheus, where Prometheus was chained to Mount Caucasus each night, and the eagle would come and devour his liver, only for the liver to return the next day. But the ancient Greeks also realized that the liver is unique among tissues. It's really the only tissue in the human body that can efficiently regenerate, and the heart, as schematized here, is completely unable to regenerate. Now, it's important to keep in mind that there's nothing intrinsic about the heart as an organ that prevents it from regenerating, because some organisms have solved this problem. Simple fish like zebrafish or salamanders can regenerate many parts of their bodies. Many of you know if the salamander loses its tail or its limbs or even a major part of its heart, it can completely regenerate those structures back to absolute normal. Our group over the past several years has asked whether there might be some regenerative window that exists in the mammalian heart, and if so, maybe we could exploit that window to expand it and reawaken it in the adult heart under conditions of myocardial infarction. And I'm not going to go into this data except to mention it in passing, but recent work from our group and our colleagues has shown that indeed the neonatal mouse heart, soon after birth, if a large portion of the heart is amputated, it can completely regenerate that heart by creation of new heart muscle cells, which restores cardiac function, and there's a lack of scar formation in this animal. But importantly, this regenerative window closes during the adolescent period of the mouse, and it never reopens again. So in adulthood, we're currently working to try to ask if we can reactivate this pathway. So, as I've said, the holy grail of adult cardiac biology now is heart regeneration. Are there strategies that could be exploited, either biological, chemical, or mechanical, that could enable the heart to repair itself following a myocardial infarction? With respect to the biological strategies for heart repair, there are two approaches that have been widely pursued. One has been extensively pursued over the past decade, and that is to use stem cells or heart progenitor cells and to introduce those cells directly by injection into the region of the scar of an infarcted heart, and to ask whether those can repair the heart. And there have been clinical studies in humans now for over a decade by many groups, and one of the leaders in this field has been Dr. Jim Willerson here in Houston. But I think it's fair to say that these approaches have encountered a number of hurdles. They're not necessarily insurmountable, but these are issues that have had to be dealt with and are still being dealt with. And they include that this is a technically cumbersome process. One has to isolate cells, grow them up in the lab, and reintroduce them surgically into the heart. It's inefficient. We now know that cells injected into the heart are not maintained in the heart long term, and essentially, even in human trials, it's been almost impossible to detect substantial numbers of cells after a month or so following injection. So there's poor survival, and cells that are derived from embryonic stem cells can cause tumors. And so it's unlikely that the FDA will authorize anytime soon the use of embryonic stem cell derived progenitors for heart repair. An alternative approach that we've begun to explore is to use cells that are resident in the heart and ask, can we reprogram those cells or divert them towards a cardiac fate, either chemically or genetically, as a strategy for regenerating new heart muscle? Now, there are two major cell types in the heart. These are essentially the bricks and the mortar of the heart. The bricks are the cardiac muscle cells. These form the structural units of the heart. They're tightly interconnected, and they beat once per second in most of our hearts. And they're separated by a type of cells called fibroblasts. And the fibroblasts are essentially like the mortar in the wall of the heart, but when the fibroblasts get activated in response to injury, they are responsible for fibrosis and scar formation. It's estimated between 50 and 70% of the cells of the heart are fibroblasts. Cardiomyocytes are among the most fascinating cells in biology. Here you can see a movie of cardiomyocytes that have been put in a dish. These cells were born to beat. It's in their DNA. You can take these cells out and they will beat by themselves intrinsically once per second. And understanding the molecular underpinnings and the mechanisms that can take a naive cell and transform it into a cardiac muscle cell is really obviously a fascinating question that many people in this room and our group are still trying to solve. It's become apparent over the past decade or so that the genes and the mechanisms that establish the identity and control the formation of the heart are ancient. These genes have existed for over 600 million years, and they function almost the same in fruit flies and mice as they do in humans. Bob Schwartz has been one of the pioneers in studying those genes and those mechanisms. And we know from work by our group and others, including Jim Martin, who's here, that these regulatory genes bind DNA, and their function is to turn on other genes. And they actually function in a network, much like an electronic circuit inside a radio or television, with all sorts of positive and negative feedback loops. But the ultimate output of the activity of these genes is that they turn on other genes that give rise to how the heart forms. And so we've asked, can we take any combinations of these ancient cardiac genes and introduce them into fibroblasts, the interstitial cells of the heart, and might those cells be able to coax those cells towards a cardiac-like cell fate? And in work that was recently published, we ultimately identified a combination of 4 gene regulatory proteins shown here. They're called GATA4, HAN2, MEF2, and TBX5. We'll just call them GHMT for short tonight. And these proteins, one of which, MEF2, was co-discovered in our group by Jim Martin, and HAN2 was also discovered here in Dallas. These factors, when introduced into many types of fibroblasts, can convert those cells into cardiac muscle cells. And recently, Deepak Srivastava's group in San Francisco has shown that 3 of these factors can also evoke this activity with lesser efficiency. When one introduces these 4 gene regulatory proteins, GHMT, into a fibroblast by putting them in a virus and infecting the fibroblast so these proteins get expressed, this is what you see. This would be a plate of fibroblasts, which skin cells we used here in a dish. They don't express any heart muscle proteins. If one introduces GHMT into those cells, they turn on muscle proteins, shown here by staining in red. And at high magnification, we see efficient activation of these proteins. Many of these cells, but not all, form intracellular structures called sarcomeres, which are the fundamental mechanical units of contraction, and others have more of a diffuse cytoplasm. This happens to be a fortuitous cell we found that has a heart-shaped nucleus in response to these factors, but they don't all have that shape. If you focus your attention here, in this experiment we took cells from the tip of the tail of a mouse. This was about as far away as we could imagine getting from the heart. These are called tail tip fibroblasts or skin fibroblasts. If you put them in a dish and infect them with a virus encoding GHMT and let them sit for about a month, they will ultimately start to beat, and I think you can see it's really wondrous to look through a microscope and see cells that typically are just some basic fibroblast, and suddenly they've been transformed into a cell that can contract almost indefinitely once per second. And so we are really dedicated to try to understand how this process might work, and can it be exploited therapeutically. Based on these and other data, this was our ultimate goal and our ultimate question. We wanted to ask, if we induced a heart attack in a mouse by tying a basically we tie a string around the coronary artery that feeds the left ventricle, causes these cells to die and fibroblasts to get activated. Could we inject GHMT into this region with a virus, and if so, might it result in the generation of new heart muscle cells inside the context of the native heart? Again, the heart, as you know, now has two major cell types, and in order to do this sort of experiment, one needs to do what's called a lineage tracing experiment. That is, one would mark the fibroblast genetically or with some stain that you could visualize it. Then you would introduce these factors and ask, can these stained cells be turned into cardiac muscle cells? So in our first set of experiments, we used a genetic marker called FSP1-LAC-Z, and this is expressed specifically in fibroblasts of the heart, and when it's expressed there, it turns them blue. But this gene is never expressed in heart muscle cells. So we asked, could we generate blue heart muscle cells by putting a GHMT into these fibroblasts? This is a bit complex, but here's how the experiment's done. One induces a heart attack in a mouse and then injects retroviruses encoding GHMT. In a normal heart, only the fibroblasts are blue with this marker and the cardiac muscle cells are not. In response to a myocardial infarction, the fibroblasts proliferate and they're activated and they form the scar and the fibrosis at the same time as the heart muscle cells die due to depletion of oxygen. The question was, could we introduce these viruses encoding these four cardiogenic factors, and if so, could we detect blue heart muscle cells, which would most likely have been derived from the pre-labeled fibroblast? This is a typical experiment. So this marker is not expressed above background levels in a normal heart. This is a cross-section through a heart. In this case, the heart is being cut this way, so you can see the chamber of the left ventricle, and here's the heart muscle cells at high magnification. If we injure the heart by a myocardial infarction and inject just a control protein called green fluorescent protein, or GFP, you can see that only the fibroblasts, these cells that are expanding in the region of the infarct and the scar, are labeled because this is a fibroblast-specific marker, as I've told you. But if we inject these hearts with GHMT, we now see intense clusters of labeled cells which correspond to heart muscle cells, and the simplest interpretation is that these cells must have been derived from fibroblasts that express the marker, because this gene is not normally expressed in a heart muscle cell. This is a high magnification view. Here you can see a cluster of labeled heart muscle cells with this fibroblast-specific marker, and if we stain with a separate heart-specific marker, you can see that each of these labeled cells is indeed now a cardiac myocyte. And without getting too complex, this is, I think, there's a couple of key points I'd like to make here. Here you can see at high magnification several cells that have been activated to, we believe, adopt the fate of a heart muscle cell in response to myocardial infarction, and here's the quantification of the number of cells with no background in this assay. One of the fascinating features of heart muscle cells is that they form what's called tight junctions, where each cell tightly interacts with the adjacent cell, and this is an essential property because they have to propagate once per second an electrical stimulus through the whole homogeneous organ, and this is mediated by these tight junctions. Here we again performed a lineage tracing experiment. So here we see a blue heart muscle cell labeled A. We believe that must have been derived from a fibroblast that expressed this marker, and it's connected to an unlabeled heart muscle cell. So we believe that's a bona fide pre-existing heart muscle cell, and right between them is this green protein shown here that connects the two. That's called connexin. That's the protein that forms tight junctions between cardiac muscle cells. If you isolate these cells through a variety of cell sorting techniques and put them in culture, this would be a normal heart muscle cell. This would be what we would call an induced cardiac-like muscle cell, or ICLM, that's been induced by GHMT. These will exhibit spontaneous contractions in tissue culture that are essentially indistinguishable from those of a native cardiomyocyte. We've wanted to ask, what about other markers of fibroblasts? Maybe this is just a unique property of this one marker, or do other markers of fibroblasts show the same lineage tracing? So we used a different gene, and in this case it labels the cells red. Again, only fibroblasts are labeled. If we inject these hearts with GHMT, these 4 cardiac factors, and then ask, can this fibroblast marker be activated in any cardiac muscle cells? We see several here that are labeled specifically with this marker called tomato because it stains cells in red. And the simplest interpretation of this result is that these tomato-positive cells must have come from a pre-labeled fibroblast with this genetic marker. And there's some aspects of sophistication of this labeling experiment, which I won't get into tonight, but some of the aficionados, particularly at the front row, might want to ask me about that later. If you isolate those tomato-positive muscle cells, which we believe were derived from fibroblasts, and put them in tissue culture, they beat spontaneously, again suggesting that they were derived from, that they are in fact bona fide functional cardiomyocytes. So the question is, if one expresses these master regulators of cardiac development in fibroblasts of a heart following myocardial infarction, can it improve cardiac function? So to do that, we repeated the experiment. We induced myocardial infarction in a mouse, and we injected these factors. Then we used echocardiography and magnetic resonance imaging to monitor cardiac function, and we found that these factors dramatically improve the function of the heart following a myocardial infarction. Here's how the experiment's done. This is a measurement called the ejection fraction. So the ejection fraction is essentially the fraction of blood that is expelled from the heart with each contraction. So mice have a very efficient heart. Every time the heart pumps in an unoperated normal mouse, about 70% of the blood of that heart is expelled. If one injures the heart by a myocardial infarction, the heart loses pump function due to loss of muscle cells, and so you can see that there's a dramatic loss of ejection fraction down to only 25 to 30%. Following injection of GHMT into these hearts, we've seen a dramatic restoration of cardiac function that's sustained in these experiments, which were measured by magnetic resonance imaging. We know that this function is retained for at least 12 weeks, but these have been carried out now for 6 months with long-term maintenance of cardiac function following introduction of these viruses. We've also looked at histological cross-sections, and I've simplified a lot of data here to just show you a dramatic but representative result. This is a cross-section through the heart. Here's the left ventricle. If we cut through the region of the left ventricle, this would be where the infarct would form following a heart attack. And here you can see in an untreated heart, there's a scar at that region. If one injects the infarct zone containing fibroblasts with GHMT, we see a dramatic rescue. It's not a complete rescue. There's still scar formation and fibrosis that occurs, but we believe these experiments establish initial proof of concept that it might be possible to reprogram fibroblasts in vivo with these combinations of transcription factors. And I should say that Bob Schwartz, who's here, has also carried out similar types of reprogramming experiments with a different constellation of factors and has also seen dramatic results in tissue culture. What I've told you then so far is that following injury to the heart in response to a myocardial infarction, as well as during aging and a number of other disorders, fibroblasts are activated, and their default pathway is to establish fibrosis and scar formation. We believe that GHMT, these cardiogenic transcription factors, have the ability to redirect at least a subset of these cells from the formation of a fibrotic scar to the formation of new muscle. Now, for the aficionados in the audience, I think it's important for me to point out a few caveats. The improvement of cardiac function that we have seen through the expression of GHMT in cardiac fibroblasts is very dramatic, and is much more dramatic than we might have anticipated based on the relative inefficiency of these factors in the reprogramming process, at least in a tissue culture dish. One interpretation of that result is that the environment of an intact heart with various growth factors and extracellular proteins and persistent cardiac contractility and interactions with adjacent cardiac myocytes might be more permissive for the formation of new muscle cells than might occur in a tissue culture dish. But it's also possible, maybe even likely, that these master regulatory proteins, while clearly able to switch on cardiac genes, they may have other activities in this type of in vivo setting. Perhaps they limit the growth of fibroblasts, perhaps they prevent the death of cardiac muscle cells, perhaps they stimulate new blood vessel formation. And it may be the aggregate of these biological processes that contributes to the positive outcome that we've seen. Nevertheless, we think that, as I've mentioned, we think this is strong proof of concept, and while clearly still inefficient and in need of optimization, it may represent a new approach for eventual treatment of diseased hearts. We believe then that the processes of heart repair and fibrosis are opposing, and our current goals are to discover the optimum combination of cardiogenic genes that can evoke this regenerative process in the intact heart, and to identify regenerative molecules, drug-like compounds, and perhaps microRNAs, which I'll talk about next, which might further enhance this process. In the second part of my talk, I want to talk about what is also the major epidemic of the 21st century and is the cause for most cardiovascular disorders, and that is obesity. These show the statistics. It's staggering to recognize that 20% of children and 65% of adults in the United States are overweight. And I think we all know that obesity is a major cause of high blood pressure, diabetes, and heart disease. This shows the statistics state by state, and you can see that Texas is leading the way on this, and I think this is probably not a race that Texans want to win. We did not set out to study obesity. Our goal, as I said, has been to understand the basic mechanisms and the logic behind how the heart forms and functions. The heart is really the most fascinating machine ever invented. The heart beats once per second for at least 80 years. It never skips a beat. There's no other pump, there's no other machine in the world that can function with that efficiency. The human heart beats 100,000 times a day. The mouse heart beats 1,000,000 times a day. The heart has a voracious appetite for energy. At rest, each of our hearts, all of your hearts sitting there, consume about 7% of your energy. And the heart demands constant energy input. It can never skip a beat. And it's remarkable to realize that the heart does not store any energy. In fact, it's been estimated that the heart only stores enough energy for the next few heartbeats. So the heart has to have a constant, uninterrupted connection with the energy resources in the body, and those cannot diminish, or the effects are catastrophic. So the heart is, in my opinion, the most mysterious, fascinating, and important organ in the body. It's the first heartbeat defines life, and the last heartbeat defines the end of life. We were studying the mechanisms that control cardiac contractility and the ways in which cardiac genes are regulated, as I told you, by these evolutionarily ancient transcription factors. The major protein in your hearts that regulates every heartbeat and controls the rate and the efficiency of contractility is a gene called myosin. Myosin is a large protein. It's incorporated into a complex structure called the sarcomere, which is the basic functional unit of contractility. Contractility is the basis for energy expenditure. This is the reason the heart has such an appetite for energy, is because contractility consumes ATP, the basic unit of energy in the body. And I should say, since I'm going to be talking about cardiac metabolism, that you are fortunate to have Heinrich Techmeyer, who's here at the front row. He is really one of the pioneers in studies of cardiac metabolism. As we were studying how the myosin gene was regulated in normal and diseased hearts, we made an unexpected observation that it encoded another gene inside of it, called a microRNA, called Mir microRNA 208. And this tiny RNA is expressed only in one tissue in the body. It's in the heart, and this shows that on a RNA blot. And so we wondered what might be the function of this tiny RNA, and why would it be hidden inside of a myosin gene? For those of you who haven't followed this field, the field of microRNA biology is exploding. There are at least 1,000 different types of microRNAs encoded by the human genome, and they're now being revealed as the underpinnings of all sorts of disorders, particularly cancer, cardiovascular disease, and others. And without getting too complex, I'll just summarize very briefly where microRNAs come from. MicroRNAs are encoded by DNA in the nucleus. The microRNA I'm going to tell you about, Mir208, is encoded actually by the myosin gene itself. These undergo complicated processing steps that aren't important for this lecture in the nucleus and the cytoplasm, but the ultimate effect of a microRNA is to bind to what's called a messenger RNA that codes for proteins and to inhibit the production of those proteins. So microRNAs function as specific inhibitors of various proteins based on the sequence of those tiny RNAs. Our work suggested that microRNA 208, this heart-specific microRNA encoded by this myosin gene, played a key role in heart disease, and we've published a number of papers on that. And based on that work, we speculated if we could develop a drug that could inhibit this microRNA, maybe it would have beneficial effects in the setting of heart disease. And so to do that, I, along with some of my colleagues from my laboratory, started a biotech company called Regenerative Therapeutics. This is not Dallas, Texas. Regenerative Therapeutics is located in Boulder, Colorado. This was our founding team of enthusiastic young scientists who took the chance to join this company. And our goal was to try to develop a microRNA drug that could target microRNA 208 or other microRNAs involved in cardiovascular disease. The approach we took is shown here. This is the nucleotide sequence of microRNA 208. It contains a short region that's known as a seed sequence. We designed a number of chemical modifications of nucleic acids, and ultimately, in collaboration with a Danish biotech company called Santeris Biopharma, we settled on a chemical structure called a locked nucleic acid, which is schematized here for the authorities in the audience. We injected these locked nucleic acid modified oligonucleotides, which have a complementary antisense sequence to Mir208 and inhibits activity. We injected those into mice and asked, the first question we had to ask was, could this drug-like compound even get to the heart? And if it could get to the heart, could it do anything? And we were really surprised by our results, and some of those are shown here. Here we've normalized the expression of microRNA 208 to a value of 1. That would be the level that's in a normal heart. If one injects mice with this drug, one sees about an 80 to 90% reduction in the level of Mir208 in the hearts of these animals. And this inhibition is sustained for at least 14 days, probably longer. The most remarkable and unexpected finding from these studies was that whether we injected this drug intravenously intraperitoneally or simply subcutaneous beneath the skin folds in the back. All these delivery methods were equally efficacious in suppressing the expression of miR-208 in the heart. I should say we're very excited about this chemical class as a potential drug-like entity for the inhibition of microRNAs. Our collaborators at Santeris recently reported the first phase 2b human trial with a locked nucleic acid modified inhibitor of a microRNA in the setting of hepatitis C virus. And they saw that they could suppress hepatitis C virus by over 1,000-fold in humans following injection every other week with an LNA-modified oligonucleotide. Now, our goal was to study the effects of this in the heart. But as we began to look at animals and we carried out extensive toxicology studies and other analyses to ask just what might this drug do in the heart, we saw that animals injected with miR-208 were resistant to obesity. They didn't lose weight on normal chow or normal food, and they didn't undergo cachexia, but they didn't gain weight. And you can see here we've put animals on a high-fat diet. Normal ones get fat. Animals treated with the drug are the same weight as animals on normal chow. And if you dissect out the fat pads, you can see here's extensive fat pads from this guy, and they're diminished in that one. We went back and looked at what the other effects of this microRNA drug might be on other aspects of metabolism that result from obesity, and the results were completely unexpected and surprising. You could just focus here on high-fat diet. Normal mice develop excessive adiposity. Animals treated with the drug don't get fat. Animals on high-fat diet have elevated serum triglycerides. Those are reduced in animals injected with this drug. This drug also lowers cholesterol on high-fat diet, and it reduces circulating glucose levels measured as area under a curve in a glucose tolerance test back to normal. Type 2 diabetes is, of course, a consequence of obesity. We've sought to try to uncover the mechanism by which this microRNA could regulate this unanticipated aspect of metabolism. As I said, microRNAs work by annealing to messenger RNAs that code for proteins. And the strongest predicted target for microRNA 208 is a little studied protein called MED13. And I don't want to get lost in jargon here, but suffice it to say that MED13 functions to control a large protein complex called the mediator complex that regulates a number of metabolic genes. And so this is, we think, the way in which this microRNA can intersect with the metabolic pathway by regulating this upstream regulator of metabolism. This hypothesis suggested if you could elevate MED13 in the heart, you would be expected to mimic the effects of this drug, because when you get rid of the drug, the target goes up. And so we genetically engineered mice to express elevated levels of MED13 in the heart. This had no effect on heart size or heart function. Here's a cross-section through the heart of this guy and of this guy. But surprisingly to us, the animals that had elevated levels of this one protein, MED13 in their heart, were lean compared to their wild-type litter mates. This guy's obviously really healthy. And they failed to gain abdominal fat. So we decided to do the opposite experiment. This was a cumbersome experiment. It took about 2 years to do it. This is what's called a heart-specific deletion. So in this case, we engineered mice so that they're lacking MED13, this one protein in their hearts, compared to normal mice. And if you just delete MED13 only in the heart, these animals become morbidly obese, and they really become morbidly obese. Keep in mind that in the prior experiments where we overexpressed MED13, we only overexpressed it in the heart. And in this experiment, we've only removed it in the heart. What that says, at least the simplest interpretation of that result, is that the heart is somehow influencing systemic metabolism, and Med13 is the switch point or the rheostat or the sensor in the heart that's enabling it to do that. And I won't go through all this data, but I just want to say if you delete Med13 in the heart, it not only confers hypersensitivity to caloric intake and obesity, it causes severe metabolic syndrome. Deleting Med13 in the heart elevates serum cholesterol, elevates triglycerides, elevates fatty liver, and causes type 2 diabetes, shown here in a hyperglycemic curve. This is our current thinking about how this process works. We believe that a key function of miR-208 in the heart is to modulate the expression of Med13. It's one of its major downstream targets, but microRNAs have many targets, so there are probably other targets involved in this. And the action of this drug, by inhibiting this microRNA, causes the upregulation of its target, and we think that then induces a signal to the adipose tissue to enhance energy expenditure. We believe then that the resistance to obesity, the effects on hypercholesterolemia, diabetes, fatty liver, etc., are secondary consequences of the regulation of this process in the heart. And so the key question is, how does the heart do this? Is there a signal? There must be a signal that the heart sends, and this would make sense. The heart, as I said, needs to sustain an uninterrupted energy supply, and so it would make sense that it's got to be in intimate communication with the energy reserves, which in the case of the body are the fat tissues. So when we published this paper, it went completely viral on the internet. Here's one paper that came out on the internet about scientists finding a cure for metabolism. There's tons of blogs on this paper, but some of them are funny, but I best not mention them here. So what I've told you then is that we believe that the heart is a regulator of energy expenditure, and the mechanistic basis for this process involves a counterbalance between the level of miR-208 and the level of its target, Med13. If we genetically elevate Med13, as I showed you, it causes resistance to obesity. If we genetically delete Med13, it causes obesity. And we can tip the balance the other way by regulating its upstream factor, miR-208. These experiments are obviously still early stage. They've all been done in mice. We don't know whether this will work in humans or larger animals, but obviously we're interested in this. I think these studies raise many interesting questions. Might anti-MIR therapies be developed in the settings of metabolic syndrome or other disorders? What is the basis for the heart to control metabolism of these tissues? Is there a secreted hormone from the heart that signals to the adipose, or is there a hormone that signals backwards to the heart? Can we find that? Med13, the key target I've described to you here, is expressed in every tissue of your body, not just in the heart. miR-208 is heart specific, so that aspect of this mechanism only occurs in the heart. But this Med13 has never been studied in any tissues in the body, and so we wonder whether there might be roles for it in other tissues. There's at present no human data, although there is one paper in the literature recently showing that a human pedigree with a hypomorphic mutation in Med13 is hyper-susceptible to obesity and hypothyroidism, so there may be some link there. So, in closing, what I've told you is that the heart is, I believe, at the center of human biology. It's been studied for centuries and is a never-ending source of fascination. I think there's a lifetime of work for myself and many people in this room to continue to try to understand how it works, why it works, how it does what it does. And tonight I've told you just about two early stage studies about strategies to exploit developmental biology and gene regulatory mechanisms as a strategy for heart regeneration and as an unanticipated strategy for treatment of metabolic syndrome. The work I described tonight was carried out by following people who made it all possible. Our work on reprogramming was carried out by two talented postdocs, Young J. Nam, an MD, Ph.D. cardiologist, and Kunhwa Song, a postdoc. Our work on metabolic syndrome and the role of Med13 was carried out by a postdoctoral fellow who's on the job market, Chad Gruder. We've had many wonderful colleagues and collaborators in Dallas that have made this work possible, and our work with Miragen Therapeutics has really been advanced because of two of my ex-students who are key scientists there, Ava Van Roy and Rusty Montgomery. And this work was all done here at UT Southwestern in our laboratory. So I'm going to close with one last comment, coming back to what Bob said. So everybody is fascinated about my connection to Willie Nelson, and since we're all in Texas, I'll tell you about Willie. I'm a music lover, and I play in a rock band. It's called the Transactivators. That's actually a gig we played in Albuquerque recently. And people always want to know, what is a transactivator? So I went to the source of all knowledge, the Wikipedia. And the Wikipedia says that transactivation is an increased rate of gene expression triggered by biological processes or artificial means. Transactivation can be triggered by transactivators. So Willie is a Texas icon, as you all know, and he was interested in what we were doing, and Willie had a concert, and he raised money for our research and created a stem cell chair. And we're also very active in an organization called Texans for Stem Cell Research. One of the key people, Kerry Kimmler, is here. This is a grassroots organization in Texas trying to raise money for stem cell research. We've talked to Willie about this, and we told Willie we want to make stem cells from his skin and call them Little Willies. And so he's enthusiastic about that. So this is Willie's bus. Everybody wants to know what it's like to be on his bus. I've been on his bus several times before concerts, and if I could remember what it was like, I would tell you. And Willie has a lot of philosophical quotes. He's also an avid golfer. And so one of my favorite quotes by Willie is, par is whatever I say it is. I've got one hole that's a par 23, and yesterday I damn near birdied the sucker. So I've tried to figure out what that means. I think it means that if you keep trying to achieve your goal, you'll ultimately achieve it. Or it might mean Willie has a hard time focusing on the hole. So on that note, I'd like to thank you all for coming out tonight, and I'd be happy to take any questions. Thank you. Yeah, Bill. Yeah, so Bill is asking a good question. Those animals have, I didn't show you the data, but they have an elevated metabolism, which you can measure in what's called a metabolic cage. So you can measure the CO2 production and the oxygen consumption by these animals, and it's highly elevated. We think that's the basis for their resistance to obesity. We've carried out metabolic tracer studies, so you can inject animals with radioactivity and you can monitor how each tissue is metabolizing things. And surprisingly to us, the metabolism of the heart is not changing in these animals, and the heart rate is not changing in these animals either, but the metabolism in the adipose and the liver is ramped up. And so they turn over triglyceride and they're they're consuming energy at a heightened rate. Again, we think that that's likely to be due to the secretion. We haven't proven this yet, but we think it's likely to be due to the secretion of a factor from the heart that signals to those tissues to mobilize energy for the heart. That factor could be a protein, could be some metabolite, and we're currently working to try to identify that. Yeah, AJ.
Enjoy your presentation very much. Fabulous indeed. You're honored to have you. One question, a couple of questions. One is on Med 13. It reminds me very much of the story of irisin. It's very similar to what described in the skeletal muscle. Is there any similarities, irisin-type molecules expressed in the heart or Med 13 potential regulate that? The second question relates to...
Let me answer the first one first. So Dr. Marion's asking a really good question. There was a report recently by Bruce Spiegelman, who actually gave the last lecture in this series, about a protein called irisin, which is released from skeletal muscle, and it has similar effects to what we've described with Med13. The highest level of expression of irisin in the body is in the heart. And so we have wondered whether irisin could be the basis for this, but we've not been able to obtain any evidence for that. So while I don't think irisin is the basis for that, the phenomenon is similar, and we're looking to try to identify the mechanism. Second question?
Yes, the second question relates to the introduction of those core transcription factors and converting fibroblasts. Would it be the alternative possibility that these are indeed progenitor cells, not really true fibroblasts? If indeed the fibroblast-specific protein is not truly the protein marker of mature cells, but also expresses immature myocytes.
So what Dr. Marion is asking is, how sure are we that fibroblasts are the source of new muscle cells in those hearts where we injected GHMT? And it's well known that there are other cell types in the heart, other progenitor cells that are resident there. We can't rule out that other cells might be reprogrammed by GHMT, and frankly, it wouldn't surprise me if some cells are. I don't think there's an adequate number of progenitors to account for the effect that we've seen, and the fact we looked at 2 different fibroblast markers would also argue that fibroblasts are a main contributor. Irrespective of the cellular source of the new muscle cells, from a therapeutic standpoint, the effect is quite pronounced, and so one might argue maybe it doesn't matter from a therapeutic standpoint, although it certainly is an interesting question biologically.
Yeah, I was just going to follow up on that question, which is that when you have injury in the myocardium, the mesenchymal stem cells default into a fibroblast phenotype very quickly, and would be a great candidate for who you might be infected because they actually make all of them. One of the ways you can tell the difference is that fibroblast comes from mesenchymal stem cell, at least in the first few days, it's still be expressing CD44, but it's mesenchymal marker. Did you ever look for mesenchymal markers on it?
It's a good question. My postdocs that did that experiment used a lot of cell surface markers in cell sorting experiments, and frankly, I can't remember whether they looked at that or not. One, it's a little bit of a detail, but it's probably a significant one, and that is that the retroviruses that we use in those in vivo infection experiments will only infect proliferating cells. They won't infect cells that don't divide. So we know that cardiac myocytes are not the target for the effect. It must be some cell type that's activated to proliferate in response to those factors.
To build off of one of the questions earlier about irisin, sort of build on this treatment, do you see, you're indicating that there's obviously communication between the heart and the fat. Do you see the emergence of a brown fat type phenotype?
Yeah, we've begun to look at that. One possibility is that this secreted factor from the heart induces brown fat, which is a heat-producing fat cell that can burn energy efficiently. I can't say that we have evidence for or against that yet, but we're looking at that. The question is, can microRNAs contribute to the reprogramming effect? Bob Schwartz showed me some data today that completely blew me away on this topic, but he wouldn't tell me which microRNA it was. And
How much is it worth to you?
I'm going to have to get him a few drinks at dinner, and he's going to spill his guts. I think it's a good question, and we're especially excited about that because I showed you that you can therapeutically manipulate microRNAs through the systemic delivery of these microRNA drugs. And so, of course, what we want to do ultimately is substitute these factors for drugs and microRNAs so you can just deliver them and do the process. And that's what we're working on right now. One last question.
You mentioned that the heart rate doesn't change when you give it. What about other physical characteristics, like the heart flow?
So the question is, what is happening to heart rate in these animals? And again, I don't want to get into too many details because it's getting late, but I'll make one final point on that. When we first observed the phenotype with MIR-208, we speculated that these animals seemed to be hyperthyroid. If you elevate thyroid hormone, it can confer resistance to obesity, and it can have beneficial metabolic effects. But thyroid hormone mimics have failed in the clinic because they have side effects that include accelerated heart rate and elevated body temperature. We do not see elevated heart rate or elevated body temperature in these animals where we've elevated their metabolism through Med13. I think that this pathway that we've uncovered is clearly connecting to the thyroid hormone signaling pathway, but it's connecting to it in a cardiac-specific way, and many of the adverse effects of thyroid hormone mimetics that have been seen in human trials are probably due to non-cardiac effects. And so this may be a way of bypassing the adverse effects of thyroid hormone signaling as a metabolic approach. With that, I know the hour's late, and I want to thank all of you again for coming.